Hello, my name is Sangmi Jeong, and I'm a graduate student in bioinformatics at North Carolina State University. I'm excited to share my research project with you, which focuses on multiomics analysis to investigate immune responses and microbiome dynamics associated with HIV resistance in Rhesus macaques. HIV, or human immunodeficiency virus, is notorious for causing HIV infection in humans. If untreated, HIV infection can progress to AIDS. However, there are no vaccines to prevent or treat HIV infection. To understand HIV-induced immune responses, Rhesus macaques with simian immunodeficiency virus (SIV) have been studied as a non-human primate model. We've observed that some female Rhesus macaques have been resistant to repeated SIV challenges over several years. They also registered a high dose of SIV viruses during this study. So our study aims to determine local and systemic immune responses responsible for the resistance against SIV infection. We also aim to identify vaginal and rectal microbiome biomarkers associated with protection against SIV challenge. This study was performed in two parts with a total of 15 Rhesus macaques. Their cervical biopsies, whole blood samples, and vaginal and rectal swab samples were collected longitudinally from each animal before and after SIV challenge. The 15 animals were categorized into three groups: uninfected, infected, and mock group. 11 Rhesus macaques were intravaginally challenged with a single dose of SIV MAC 251 viruses. One of them was challenged twice across both parts of the study. That's why the total number of animals is 16 in the table. And 4 Rhesus macaques were sham inoculated as a mock group. Cervical biopsy and whole blood transcriptors were analyzed to investigate local and systemic host immune responses. Figure 1 shows that whole blood samples from 3 animal groups are also separated into 3 separate clusters by their expression profiles. As shown in Figure 2, the whole blood RNA-seq data was further examined to determine differentially expressed genes regarding temporal changes in transcriptional responses to SIV challenge and the pathways involved. For example, the A set of DE genes was found to be associated with genes encoding cell cycle-related targets of E2F transcription factors. Rectal and vaginal microbiome profiles were defined using the refer region of 16S rRNA sequences obtained from rectal and vaginal swab samples. The 16S reads of microbes were processed into amplicon sequence variants, or ASVs. Here, we investigated the differential abundances in the rectal and vaginal microbiomes among 3 groups. In Figure 3, Microbiome profiles distinctly segregate into rectal and vaginal swab samples, as well as within each animal, but not into 3 separate groups. The temporal changes in abundances among 3 groups have not been investigated yet. However, throughout the time points, an ASV assigned as Porphyromonas species showed a significant difference between infected and uninfected groups in vaginal microbiomes. This suggests that the Porphyromonas species potentially serve as a vaginal microbial biomarker associated with SIV challenge. Our study is valuable as this enables the simultaneous analysis on host transcriptomes, and microbiomes, which potentially contribute to HIV-induced immune responses and HIV infection outcomes. These findings may elucidate the protective mechanisms against HIV in humans. Thank you for your attention, and please feel free to reach out with any questions.